next section, we consider our taxonomic relationship with the other mammals, and in particular primates. We start with the analysis of hominoids. And the hominoids share a number of phenotypic features with the other anthropoids. Traditional morphological classifications of primates identified two groupings: the lower primates, prosimians, which included the tarsiers and lemurs, and the higher primates, anthropoids, which included the apes, monkeys, and the humans, of course. The extant anthropoids are characterized by complete bony eye circuits and lobar molars with rounded puffy cups, and can be further resolved morphologically into three groups. The New World monkeys have 12 premolars and side-facing nostrils, while the Old World monkeys and the Hominidae, which includes humans and other apes, have 8 premolars and a nose with downward-facing nostrils. These distinctive morphological features define the Catarrhini clade, so we belong to the Catarrhini clade. On the other hand, most Old World monkeys and New World monkeys share numerous primitive traits, for example, the tail and slightly shorter forelimbs, whereas hominoids had the longer arms. The loss of the tail is the defining feature of the great apes evolution, and here it is why.
Tails are like opinions. Basically, everyone has them. Fish, birds, most reptiles, and even some of our closest relatives. So, why are we missing out? Since tails first evolved at least 500 million years ago, they've taken on every role imaginable. Geckos use them to store fat. Birds use them to steer through the air. And rattlesnakes use them to scare off predators. But for most mammals, they serve one major purpose: balance. Yet, as you get closer to humans on the evolutionary tree, tails disappear. Gorillas don't have them. And neither do chimps or any other ape, including us, of course. To understand why, take a look at how we walk. Some of us primates crouch with our chest held diagonally to the ground. Others, like gibbons and humans, can walk completely upright. Now, walking like this gives us a huge advantage because unlike four-legged animals, which have to pour energy into every step they take, two legs take advantage of gravity, which does some of the work for us. You see, each time we take a step, gravity pulls us forward. The end result is that when we walk, we use around 25% less energy than walking on all fours. And in the wild, every ounce of energy you save can mean the difference between survival and starvation. But this way of getting around also totally eliminates the need for a tail. Because even though a human head weighs a hefty 5 kilograms, it sits on top of the body when you walk, not in front. So you don't need a tail as a counterbalance. Pretty disappointing, huh? That being said, you can still see a reminder of a time when our ancient primate ancestors had one. Just look at a human spine. You can see how the last few bones are partially fused together. That's your tailbone. It's all that's left of our tail. And yes, it's sad and pathetic, and you can't wag it. Now, in rare cases, babies are born with what looks like a tail, but that's not what's really going on. Most often, these tails are actually tumors, cysts, or even a parasitic twin. Even more occasionally, they're a true outgrowth of the spine, but are completely boneless, a soft tube made entirely of fat and tissue. These types of tails usually form as a birth defect, a deformity of the spine called spina bifida. And in these situations, doctors will surgically remove the tails with no harm to the baby. But as cool as it might sound to have an extra limb to swing through the trees or keep mosquitoes away, we are who we are today because, well, we don't.
Apparently, loss of the tail was the defining event that separated clades of apes and monkeys, enabling the former to develop more versatile locomotion and more upright posture. One of the characteristic innovations found in apes and Old World monkeys is their trichromatic vision.
It's estimated that a typical pair of human eyes can see hundreds of different colors. But 23 million years ago, our primate ancestors were red-green colorblind. So how did we come to have the rich color vision we have today? I'm in Seattle to meet one of the world's experts on color vision, Jay Knights.
Oh, hey, Neil Schumann. Hi.
Great to meet you.
Good to see you. Well, great to be in the color lab.
Jay's been studying how animals see color for the last 25 years.
This is the place, huh?
Yep. This is where we test color vision in the monkeys.
Experiments conducted here show that most mammals, including many primates, are red-green colorblind.
This is Kramer.
Hi, Kramer.
And Kramer is colorblind.
Kramer is shown a red blob on a green background. If he kisses the red, he gets a treat.
If you can't see red or green, this is completely invisible.
Most humans can clearly see this red blob, but Kramer can't. He can't tell the difference between reds and greens. So why can't Kramer see the red blob?
Okay, Kramer, you were a star.
Kramer's eyes, like all eyes, rely on special proteins called opsins to detect color. They're held in thousands of special cells in the retina at the back of the eye. Kramer's got two types of opsin, each tuned to specific wavelengths of light. Signals from these opsins are then interpreted by the brain, which allows Kramer to perceive color. But to see color like we do, Kramer would need a third opsin, tuned to different wavelengths of light. We think our early primate ancestors were like Kramer. They had just two opsins as well. So how did they evolve a third opsin? The answer is in our DNA. Each opsin is encoded by a single gene. And when scientists compared these genes, they found that the gene for the newer opsin sits right next to one of the old ones. And significantly, they are incredibly similar. Both facts are telltale clues as to how the extra gene evolved. The old opsin gene was duplicated, and over many generations, one of these copies acquired small mutations that allowed it to detect different wavelengths of light. Could this change alone have really driven our ancestors' color vision? To find out, Jay set up another experiment. He implanted a third opsin gene from a human directly into the eyes of a colorblind squirrel monkey named Sam.
What we did is really a test to see what's the minimal thing you could do in order to give an animal color vision.
The results were astounding. Sam used to fail this test. Now he can easily tell the difference between reds and greens. Jay has recreated evolutionary history and given Sam human-like color vision.
You might think, oh, it would take them a long time to learn this new pattern in their brain. But as soon as the gene was turned on, the animal began to make these discriminations that they couldn't make before. And so in one, very short evolutionary step, it goes to this totally different world. You go from just having strictly, let's say 5 colors, gray, black, white, blue, and yellow, to hundreds of different colors that are all the blues and greens and purples and oranges.
One simple shift opens a whole universe of...
Yeah, that's the amazing thing. It's a multiplicative effect.
The ability to see a broad range of colors made it easier for our ancestors to find ripe fruits and young leaves. They passed their color vision to their descendants and eventually to us. We owe our rich color vision to monkeys living in an ancient forest.
The ability to distinguish green and red is due to the long wavelength sensitive LWS gene duplication on the X chromosome. The duplication event probably occurred at the start of the radiation of Catarrhines, some 30-40 million years ago, because most extant Catarrhines carry two functionally distinct copies, OPN1LW and OPN, which is opsin 1MW, of the ancestral gene, while the New World monkeys have only one copy. In contrast to Catarrhines, almost all mammals have two photopigments produced by autosomal short wavelength sensitive blue and the X chromosome long wavelength sensitive red-green genes. While the advantage of trichromatic vision include, for example, the distinction of the ripeness of the fruits, experiments on the New World monkeys have shown that dichromatism may also have some advantages, potentially in superior visual ability to discriminate color camouflaged shapes and patterns. Compared with other primates, Catarrhines are less able to rely on smell as a cues, and this may be because of their improved vision. More olfactory receptor genes have become pseudogenes in those species that have trichromatic vision than in dichromats. One of the most important questions in the human evolution is the phylogenetic relationship within great apes clade. Albeit humans are the most morphologically divergent among the apes, the phylogeny cannot be resolved using only morphological traits. Like the morphological features of the animals themselves, the morphologies of their chromosomes provide information about the relationships between primate species. The number and physical appearance of mitotic metaphase or late prophase chromosomes under the microscope is called the karyotype. Because of their relative stability, comparisons of karyotypes among the higher primates can give quite robust information about branching order. However, no cytogenetic clock is available to calibrate the gross changes that chromosomes undergo, so karyotypic analysis gives no information about the dates within the phylogeny. For the dates, we have to use genetic sequences. The most obvious difference between the chromosomes of humans and the great apes is their number. While humans have only 46 chromosomes, chimpanzees, gorillas and orangutans have 48. Comparison of G-banded karyotypes, however, demonstrates a very high degree of similarity between the four species and shows that the difference in chromosome number results from the apparently simple end-to-end fusion of two small chromosomes seen in the great apes to form chromosome 2, and they form chromosome 2 only one in humans. One centromere was inactivated. In the quiz, we will compare the chromosomes in order to support one of the human chimps, gorillas, orangutans phylogenies, built using only morphological observations. In summary, in these parts, we considered the following topics. Humans and great apes can pass the pathogens to each other, thus driving evolutionary response of each species. We overviewed major differences in brain structure of chimps, humans, and touched on the differences in the genes which differ. More about this topic you can read in this very accessible review of genes affecting the brain, which makes us different. Morphological evidence from fossils and extant species places humans in the taxonomic order of primates and reveals humans to be most closely related to the chimpanzees, bonobos, gorillas and orangutan. The karyotypes of humans and great apes are similar but different in a number of respects. The most notable being the reduction of chromosomal number from 48 to 46 as a result of a chromosomal fusion of the hominin lineage. The time since humans and great apes last shared their common ancestors can be estimated from molecular comparisons between species. These calculations can be reconciled with fossil-derived estimates of the timing of the speciation events. Genetic diversity in the great ape species appears to be significantly greater than in humans, despite they are now having much smaller and decreasing population sizes.